My name is Roland von Bothmer. I'm a retired professor of genetics and plant breeding on the Swedish University of Agricultural Sciences. It's really a privilege to be elected chair for this morning for three fundamental lectures on plant biology. So as introduction, we have three world-leading scientists for this, and we start with Ottoline Leyser, University of Cambridge. And you have been working with genetics of plant and plant development and interactions of plant hormones, especially auxins, with the environment. So please. And the title of your lecture is Genetics from Mendel over McClintock to today. So please, Ottoline, and you have 45 minutes. And if there are some spare minutes, I can allow if you wanted 2 questions after each lecture. So please.
Good. So thank you very much for the introduction. It's really a pleasure to be here, to be able to talk about one of my favorite things in the world, and also hopefully to contribute to one of the things I think is extraordinarily important in the world, and that is the relationship between science and everything else. And that means science in society, its position in society, and also its contribution to policy. So I'm here as a plant geneticist from Cambridge, but I also spend quite a lot of my time in the sister organisation of the Academy here in the Royal Society in London, where I chair the Society's Science Policy Advisory Committee, so the work we do on science for policy and policy for science. So it's a pleasure to be here as part of this effort. So as I say, what I'm actually going to talk about, though, is one of the things I love most in the world, and that is genetics. I'm a plant geneticist, and the reason I find genetics so extraordinary as a discipline is because it does this extraordinary scale bridge. And I would like to illustrate that with some pictures that were mostly stolen from my colleague Enrico Cohen in Norwich, but I think they really capture this extraordinary thing that is at the core of genetics as a science. So here is my version of the picture of Araby Topsis. It's this little thing here hiding in the grass. What you've seen already was the flowering stem. These are the leaves that are made before flowering in the rosette. And so here they are, sitting in the grass, and the whole thing is a couple of centimetres across, maybe a bit bigger. And if you start to zoom in, down the orders of magnitude of scales, then here's a leaf. And if you zoom into the leaf, you'll get to the point where you can begin to see the cells, also that we've heard about already, originally described by Hook. And you zoom in a bit further, and actually the cells on the Araby Topsis leaf surface has this extraordinary wiggly jigsaw shape. They're very beautiful. And then if you are able to get inside those cells and look at those with an electron microscope, you begin to see the structures inside the cell. So here's it's a fracture through the cell, and what the red square is now on the nucleus where all the DNA is, and one can zoom in on the nucleus and see that there are nuclear pores, and then that connect the nucleus to the outside areas. And then into the nucleus you can see here by now a diagram of the DNA, which is wrapped round and round proteins to form these fibers. But then if you zoom right into the DNA, you can see the famous double helix, and across the 2 helical sugar backbones, the bases are bridging in like a kind of twisted ladder, and it's the order of just those 4 bases that most people know about, A's, G's, C's, and T's, that encodes the genetic information. And you'll see by now we're on 10 to the minus 9 meters, so in the nanometer scale, and we started off way up here at the kind of centimeter scale. So we've bridged 8, 9 orders of magnitude. And that's the bridge that genetics does. The genotype, that's the information in the DNA, the order of those A's, G's, and C's in the DNA, that's at the order of nanometers, but it's influencing things at the order of centimeters, or if you're a big organism, meters. So across those 8, 9 orders of magnitude, tiny, tiny changes in the DNA have big impacts on what you look like as an organism. And just to put that into perspective, that's about the same as the relationship between those centimeters and the entire planet going in the other direction. It's an extraordinary thing that those tiny nanoscale events are influencing across multiple orders of magnitude what we look like. And genetics is an extraordinary way to bridge that gap, to understand the relationship between those things. Because at its core, it involves understanding how those single tiny changes in the DNA that maybe just change one of those bases out of the thousands and thousands in the genome of organisms can influence what the organism looks like and how it behaves. And then our job as geneticists, particularly in my field, which is developmental genetics, is to try to fit in all the stuff that goes on in between those two. That's a much bigger job, but nonetheless, the fact that we have these genetic tools has been completely transformational in our ability to do that. And it all started, as we've heard already, with Mendel. And I am going to use exactly the example that you mentioned of these round and wrinkly peas, because it provides a fantastic example of how you can really span that gap, understand how one tiny change at the DNA level results in a change at the level of the organism, which we all recognize from our high school biology. And this is the story of Gregor Mendel and starch branching enzyme. That's maybe something you haven't heard of, but it turns out to be absolutely crucial to what you have already heard of today, which is the round and wrinkled pea trait that was one of the early traits that Mendel worked on. So here is a pea pod, and in the pea pod you can see that there are, some nice, smooth, round peas like this, but also there are some wrinkly, ugly-looking peas like this. And it was this trait that was one of the core traits that Mendel followed through the generations to try to understand how traits were inherited between generations. So everybody can look around them in the world and see that you resemble your parents in some way. So something is passing from you to your children, and that is the kind of hereditary idea that is kind of obvious, but how does that work was what Mendel was interested in doing. And his key innovation really was to use plants, which was really helpful because plants can be inbred, and so you can inbreed them for many, many generations, and you wind up then with genetically very homogeneous lines that are absolutely kind of clean, and he called those true breeding. So if you go through the generations, you can get to the point where the offspring look exactly like the parents every generation. So you start with true breeding peas, and he found lines that were true breeding for that round seed trait and lines that were true breeding for the wrinkled seed trait. So every single one of their seeds was either round or wrinkled in that context, and then he crossed them together. And in the next generation, what he found was that all the peas were round. So despite the fact that one of the parents had the wrinkled trait, all the peas in the next generation were round. And then if he took those next generation plants and crossed them between themselves, amongst each other, then, or cross them to themselves, that's another way to do it, then in the next generation after that, then you get the very famous 3-to-1 ratio, which I'm sure is familiar to some people if they remember back to their high school genetics. And that's 3/4 of the peas were round, but 1/4 of them were wrinkled, so this wrinkled trait re-emerges in the second generation, and you can see it. And in fact, this original pod I showed you here, if you count, you really get exactly 3-to-1, 3 times more round peas than wrinkled peas. It's statistical, but in this pod, it happens to be exactly 3-to-1. So just based on looking at these phenomena and thinking about them, Mendel came up with these laws, and I'm only going to talk about one of them, and that's the first one. And what he argued was that traits were governed by some kind of hereditary factor, a unit that we have later termed as a gene, and that the genes exist in pairs. So you have a pair of every single gene in you, as do the peas, and those pairs, one is inherited from your mother and one is inherited from your father. And they don't mix in life, he argued, and then they segregate, so they're passed randomly into the next generation. So the idea then is that this true breeding round pea has two versions of this gene that make your peas round, and the true breeding wrinkled peas, which is written little r for reasons I'll get to shortly, it has two versions of the gene that make your peas wrinkled, and in the next generation you get one of each of those. So you get the round version from your one parent and the wrinkled version from the other parent. So absolutely all the children have that one of each thing. And that turns out to result in a round pea, and we'll get to that in a second. And then if you think about this plant, segregating those into its gametes, into its sperm and eggs, the sperm and the eggs only get one of those, and they randomly either get the round version or the wrinkled version. So now you're back to half your gametes being round, half your gametes being wrinkled. And if you cross together two organisms that are like that, so you just randomly mix up those things, then you'll get a quarter will get two rounds, a half of them will get one round and one wrinkled, and a quarter will get one wrinkled each. And as I've said, if you add to that this concept of dominance, which is another important Mendelian idea, and you argue that if you have this round version of the gene, you can't see the effect of the wrinkled version, so all of these guys will look round, then you get this 3-to-1 ratio out of the fact that all of these are round, whereas this one looks wrinkled. So that's the core concept that Mendel came up with, which was absolutely extraordinary in many ways, to reduce what looks like an incredibly complicated business to this key idea of hereditary factors that exist in pairs, one from each parent, they don't mix in life, and then they're segregated separately to the next generation. And we use this today all the time in the lab, and it's still super important. And I'm always very impressed by the fact that this was going on before we had any idea about DNA. We had no idea what the genetic material actually was. And it then has become a very interesting job over the past 150 years or whatever to try to figure out how this works physically, how these concepts about hereditary factors that don't mix in life and pass from generation to generation, how those concepts translate into what we know about chromosomes and DNA and genes and all of these kinds of things. And to cut an extraordinarily long story short, we know now that genes are made of DNA and that the DNA are on chromosomes. So these, we have 23 pairs, and you get one of that set from your parents and from your mother and one from your father, and different organisms have different numbers of pairs. Araby Topsis, as you've heard about, our favorite plant, has 5 pairs. And the behavior of those chromosomes is exactly the same as the behavior of the genes, in that they arrive, they don't mix in life, which is a slight oversimplification because they do a bit, but I'm not going to talk about that. And then they segregate into the next generation, and they carry that genetic information in exactly the same way that Mendel saw, and that's the so-called chromosome theory of hereditary. And then we're then able to work out that it was the DNA in the chromosomes rather than all the protein that's in the chromosomes that does the job. And then the trick is to try and figure out which of those genes, the many genes that are on the chromosomes, could be responsible for this round and wrinkly trait. And to cut an extraordinarily long story short, we now know it looks something like this. So it turns out that those wrinkly peas have more sugar and less starch than the round peas, because they're missing an enzyme, which we've also heard a little bit about already, which is a protein that catalyzes a particular reaction in the cells that allows starch to branch as it's being made. I'm terrified because I have a starch expert or a sugar expert in the audience, and it's not my field. So Mark will leap in and correct me if I get it horribly wrong. And because it's lacking that enzyme, it's not as good at making the starch, and so there's a sugar kind of left over in the seed as it gets to the end of its production time. And because there's sugar, they then suck in water by osmosis, and then when they dry out, they lose the water and they wind up wrinkly. So the reason those peas are wrinkly is because they can't make starch very well. They overaccumulate sugar, and that results in them being full of not just carbon, but also water at the time that they have to dry out, which they lose, and then they get wrinkly. It's quite simple, really, and it's why it's quite such a nice, powerful example, because it's a relatively short chain of events that takes you from the gene, which encodes this enzyme called starch branching enzyme. That gene in the cell would be copied into an RNA, messenger RNA molecule that allows the information in that base pair sequence to be decoded into a protein, and that protein is starch branching enzyme, and that starch branching enzyme allows starch synthesis to happen. And if you don't have that gene, if that gene is defective, which is what goes on in the wrinkly case, then you don't make the messenger RNA, you don't make the protein, your starch synthesis is slow, so there's less starch and more sugar, because the way starch is made is by sticking all the sugars together, and then there's more water, and then and all of that kind of stuff. It's a very short chain of events, and it links these nanoscale events up to the centimeter events in the seed, very simply and elegantly. And I think the other thing that is quite powerfully illustrated by this example is that it very clearly explains this dominance business. Dominance sounds a bit mysterious. You have to arbitrarily invent the fact that the round version somehow stomps on the wrinkled version, and if you have one round version of the gene and one wrinkled version of the gene, then you're going to be a round P. But if you know that the wrinkly phenotype is essentially caused by a change in the DNA that breaks the gene so it doesn't work anymore, so it's now not encoding anything at all, then it makes quite a lot of sense, because in this round, wrinkled, so-called heterozygous version, where you have one round version and one wrinkled version, there is the one round version. The round version is perfectly good at making the enzyme, and just one copy turns out to be enough to do the job fine, and so the P is round. And it's not unless you have no versions that can make the enzyme that you begin to see the effects in the phenotype. So without having any idea, it turns out that Mendel was working on starch branching enzyme, and it's taken us a number of years since to figure out that's what was going on. But because of the extraordinary tools we have from molecular genetics now, it's much easier to move to and fro between a phenotype, so what the plant looks like, back down to a genotype, so the underpinning change that influenced that phenotype in both directions. So we can ask, what does this gene do by on purpose breaking it, as I'll get to, and seeing what effect that has on the phenotype, or we can look for genetic variation that causes a phenotype that we're excited about, and then figure out what the genetic basis is underlying that, what the gene changes are that drive that. That's the core of modern molecular genetics that has completely revolutionized our understanding of how plants work and given us the tools to drive, for example, plant breeding in a much more directed way, which I'll get to later. But rolling back a little way to the kind of middle of the last century, as all of this work on genes was emerging, the idea that came out of that really was that genes were maybe a bit like beads on a string. You had these hereditary particles that were considered to be quite kind of stable, unitary things, and you could think of chromosomes like a whole kind of lineup of those beads on a string that were contributing to the traits. There were lots of them. They could be quite complex, but nonetheless, they were units, solid units. And I would say that as well as this connection between the levels of scale in biology that has really powered up over the last few decades as a result of molecular biology, the other really major change conceptually has been in our thinking about how genomes work and what genomes look like. Because here, I think the early ideas of Mendel have been completely revised and overturned, whereas the big concepts of the relationship between genes and what organisms look like are absolutely rock solid and the basis for everything we do today. This notion of the sort of stability of the genome has gone long since out of the window, and one of the reasons that it's done that is because of the work we've also heard mentioned today of Barbara McClintock. So I just have one slide that encapsulates her work, and she used the same tools, the extraordinary power of Mendelian genetics, working before we knew that it was DNA that was the genetic material, to demonstrate the instability of the genetic code in a way. This notion of the incredibly solid, don't mix in life, nothing changes, she threw away by demonstrating all kinds of things, and most powerfully, perhaps, as we've heard, that there are bits of DNA, which she didn't really know were bits of DNA, she called them controlling elements, that actually moved around the genome. And the way we now think about those is we call them transposable elements, and they are indeed sections of DNA that either can copy themselves, and the copy goes and sticks itself somewhere in the genome, or they can cut themselves wholly out of the genome and go and stick themselves back in somewhere else. And these transposable elements turn out to be incredibly prevalent throughout the genomes of all kinds of organisms. And there is an interesting discussion about their relationship to viruses, because most of them are quite closely related to viral genomes. And a general idea is that either transposable elements evolve from viruses, or viruses evolve from transposable elements, or some kind of combination of those two things. And they are essentially parasitic in your genome, although then the genome has harnessed them to do useful things. Again, this notion of purpose is quite problematic, because you've got to worry about the selection acting on the viruses or virus-like things in your genome, but also selection acting on the organism that's hosting them. And both of those things come into play to maintain and change the number of these elements there are in the genome. So, okay, so how can I convince you that such things exist? So it took Barbara McClintock a very long time to convince anybody that such things existed, but she did eventually do it, and as we heard, eventually won the Nobel Prize, because it was such an extraordinary concept in people's mind, because they were so fixated on the sort of solidity of the beads on a string idea in the genome. And she worked on maize, and she worked on these kinds of speckly maize that provide rather curious phenomenology in the context of phenotype, which in this case is whether the seed looks yellow or purple. And so she worked a lot on alleles or versions of this gene that allowed seeds to look purple. And she found unstable versions where in some of the seeds, as you can see here, there's this kind of speckly appearance where most of the kernel looks yellow, but some of it looks purple. And these have become quite trendy, actually, in terms of making fancy corn chips, and you can buy corn chips in weird different colours these days. But possibly people are more familiar with this kind of speckly appearance if you have a garden and you have snapdragons. There are quite a lot of varieties of snapdragons that also have this kind of speckly sectored appearance. And the reason for these things is because there is one of these transposable elements sitting in the middle of a gene that allows the plant to make an enzyme that makes this purple pigment, and if it jumps out of that gene, then it will restore the normal purple version, and you'll get a little purple speckle. But if it stays in the gene, the plant will be unable to make that enzyme that makes the purple pigment, and so it will be either yellow in the case of the kernel, or this white background as in the case of the flower. And so this kind of kernel, that kind of jumping out event happened really early, so you get the entire kernel looking red. This kind of kernel, it didn't happen at all, so the entire kernel is yellow. And in these kernels, then there were multiple events of jumping out quite late, so you get a little patch of red compared to most of it being yellow. And these transposable elements, as I say, turn out to be just astonishingly prevalent because of their, almost certainly because of their kind of parasitic nature. And indeed, as we also heard mentioned earlier, it transpires in a lovely twist of fate that this original mutation, the round and wrinkle mutation, is caused by a transposable element sitting in that starch branching enzyme. It happens to be a very inactive one that doesn't tend to branch out very much, jump out again very much, which is why Mendel was able to use it as a stable trait in his experiments. Now, we've also heard about the extraordinary amounts of DNA that some plants have, and indeed some organisms have. There's an incredibly wide range of amounts of DNA in different organisms. This does not relate in any way to the number of genes they have, and that's because the vast majority of the DNA in most organisms, particularly organisms that have a lot, turns out to be not genes like starch branching enzyme, but rather these kinds of repetitive elements, which include transposons, which tend to amplify themselves up during evolution. So an illustration of that here, this is a diagram of the DNA in the wheat genome, which has fairly recently been sequenced. This tiny yellow bit at the top, okay, so wheat, I should say, is one of these hybrid organisms. It's actually got 3 genomes, and so that's what these 3 bars are. They're the various 3 genomes that have been kind of amalgamated over the years of domestication. And the 3 genomes each have some actual genes, and that's this little tiny yellow bit on the top. Those are the genes that code for proteins. It's a tiny fraction of the total amount of DNA, and the vast majority, all of these coloured bits, are various kinds of transposable element that are more or less active, that have been amplified over the years and just sitting there clogging up the chromosomes. There's a massive debate about how useful they are, but they're there. They're definitely there, and they are a tiny fraction. I mean, the genes are a tiny fraction of the total genome, and a huge amount of the genome is made up of this DNA that is quite unstable and involves things jumping around and all the rest of it. So I like to say to people that if you had cornflakes for breakfast, it is almost certain that at least one of those cornflakes had a bit of DNA stuck in it at some new place that no one has ever eaten before, because it's not that transposition in cornflakes is very high, but there are thousands of transposable elements, and one of them will have moved into some new place in the genome. The idea of this sable, lovely beads on a string thing is gone. Genomes are a mess, and somehow they work. And it's amazing, and we're very happy with that. And indeed, were they not a mess, we wouldn't be here, because the mess, this extraordinary churn in the genome that is what drives evolution. Okay, so let's move to that. Genetic variation, which you can also call mutation, but mutation sounds scary, whereas variation sounds good, but it's the same thing. Genetic variation or mutation comes from many, many sources. If you think about all those millions of base pairs that need to be copied every time you divide your cell, there are going to be mistakes when that happens, which will change the order of bases. There are those transposable elements, which are most of the genome, that go around wreaking havoc. There are quite big rearrangements in the genome that happen fairly regularly because, for example, the transposons quite often will jump out. The genome isn't stuck back together very well, so then you've broken it, which is also quite a common event. And sometimes it repairs wrongly, so huge bits of the genome can move around. You can have environmentally induced DNA damage. We all know about that from for example, the causes of skin cancer, which is UV radiation, often from the sun, and that's going on all the time in all organisms. When you pass your chromosomes down to the next generation, the part about not mixing in life is partly an oversimplification, because it turns out you get swapping between the chromosomes from the mother and the father in a process called recombination that mixes things up. And in repairing those swappings, you can quite often get mistakes. The DNA can be damaged. There are lots of repair mechanisms, but sometimes those repair mechanisms don't work very well. And then there are these much bigger things, which we've heard a little bit about before, like whole genome duplications, where the separation doesn't work properly during the formation of gametes, and so you wind up with double the genome. That's quite common, particularly in plants. And hybridizations between closely related species, which also amplify up your total number of genomes, is quite common. And the wheat example I gave you earlier, wheat is actually a hybrid between 3 different grasses. And the reason that strawberries have such a ridiculous number of genes is because they actually have 8 genomes. So because of these kind of duplication events rather than hybridization events. So in my view, they're kind of cheating. They're just it's 8 times counting rather than having 8 times as many genes. But anyway, that's how it works. And then there is horizontal gene transfer. So those viruses I talked about, which are related to transposons, there are plenty of viruses that infect cells that stick their genomes into the DNA of the organism, and then they will excise it out again and take it off to another thing. And you can move genes around that way, even between species. And a lot of plant biotechnology is based on some of those kinds of organisms, and I think Mark might or might not mention that later. So one of the ones we use a lot in plant science is a soil-living bacterium that sticks DNA into plants, and there is good evidence that that has actually influenced the evolution of some plant species. So lots of sources of genetic variation arising from this ugliness of genomes in many ways. And it sounds scary, but if it hadn't happened, there would be no evolution. So the difference between us and a chimpanzee is actually a surprisingly small number of mutations, but the difference between us and a carrot is a rather large number of mutations, but nonetheless, ultimately, most of the evidence suggests only one origin of life on Earth, and so the difference between us and some kind of single-celled simple thing, a whole bunch of mutations. So what's driven then this variation and evolution in general is this process of natural selection. We've also heard very briefly about Darwin. My research institute, where I have the pleasure to work, is in the Botanic Garden in Cambridge, which is credited as being, in many ways, the kind of root of a lot of Darwin's thinking about natural selection. It was founded by John Henslow, who was Darwin's mentor, and he set out the garden in a way that is the beginnings of the thinking about the relationship between different plants and their evolutionary common ancestor type origins. So and Darwin, definitely a plant scientist. In the basement of our building is a herbarium, and many of the specimens in the herbarium were sent by Darwin to his mentor, John Henslow, when he was on his journey, his voyages of the Beagle. And so he figured out, I suppose, that the driving force behind evolution was this concept called natural selection, where if some mutation or variation in your genome arose, and it was a variation that resulted in you being fitter, in other words, having more children, or rather technically speaking, more grandchildren, then that mutation would help you to do better than your neighbors, and your genes would then be the ones that were overrepresented in the next generations, and so on and so forth. So you could drive changes. You could get from a common ancestor between chimpanzees and people, both to chimpanzees and to people, by mutations in the common ancestor, resulting in different phenotypes, which I've talked about, and those phenotypes give different advantages and disadvantages to the organisms that carry them, and they then survive or don't survive in particular environments, and so on and so forth. But crucially, Darwin also worked out that natural selection has a cousin, and its cousin is domestication. And that's where we, as humans, have taken organisms out of the constraints of natural selection and instead put them under human selection and decided which things we think are the best. So that same mutation also underpins domestication, and so all the different breeds of dog we have a common ancestor in a wolf-like organism. And so as a result of human selection rather than natural selection, all of this diversity in dogs has arisen, underpinned by mutation, exactly that same long list of processes. So this you might call the survival of the favorite rather than the survival of the fittest. And a crop plant is very much in that category. A crop plant is a plant where natural selection has been replaced by human selection for the driving force in its evolution. So survival of the fittest becomes survival of the favorite. And this makes a big difference to the way that we need to think about both the whole concept of natural, to be honest, but also the process of crop breeding. So natural selection has driven the selection of individuals that survive best in the face of competition. And so formally, the definition is to do with how many grandchildren you have. So you're fit if you have a lot of children, and those children survive really well to produce grandchildren. And so that's the definition of success, effectively. And so you're going to allocate your resources to maximize the number of offspring in the face of all this competition. So you're making all the time, as an individual organism, or in this case a plant, decisions about what to do with the carbon you have, what to do with the energy you have, what to do with the nitrogen you have, the limiting resources you've collected, in order to ensure that you have the most grandchildren. That's what evolution has done. It's not an optimizing process. It does its best with what you've got, but it is a slave to historical accident. And I think we might hear a little bit about that later, because if you think about it, there are things that were, all you've got to go on with selection, or the only thing you can improve is what you've got already. What you've got already in evolution was selected quite often for something completely different. So evolution is done an extraordinary job, if you think about it, in driving diversification and in creating organisms that can survive in all kinds of landscapes, but it is not the best. And even the extent to which it is the best is driven by the fact that the driving force for it is maximizing the production of grandchildren in the face of this highly competitive environment. So what's evolved are complex dynamic regulatory systems that support that goal. And for plants, that tends to be a very flexible way of growing, so you can tune your growth according to the environment in which you're growing. And then all kinds of things, for example, that defend you from attack. And particularly the seed, which is your children, needs to be very strongly defended, so it tends to be indigestible or poisonous. It's also spread widely from the plant, because that's a very successful strategy. And the idea is to maximize the number of grandchildren you have, to want to make as many seed as possible with the minimum resource they need to survive. And these kinds of traits, I just picked a few of them, are actually hopeless from an agricultural point of view. So agriculture, you want to select plants that maximize resource allocation to the part we want to eat, which is not necessarily the part that the plant thinks is a good idea, as a way to put it. You're not interested in the plant competing with its neighbors. In fact, you really want it to play nicely in the field. You've got rid of as much of the competition as you can, and the only other plants growing next to it are other plants you want to thrive as well. So no competition, please. And to do that kind of adaptation, you need to modify these systems accordingly, and we like this a less flexible growth habit. A lot of this, the flexible growth that plants have is about competition, which we want to reduce. And then, of course, all of these seed traits, mostly what we eat is seeds. We'd like them to be digestible and nutritious, and we don't want the seed to be flung off the plant. That would be really annoying for harvest. And, of course, we want the seed to be big and juicy and maximally resourced. And a really good example of the kinds of things that domestication has done is captured on this slide. This is teosinte, which is a very close wild relative of maize, and it looks like this. It has got the small, indigestible, minimally resourced seed, and 8,000 years of Mexican farming plant breeding ingenuity has resulted in maize. These two species are actually still cross-fertile, so we can do the genetics. We can, in the Darwinian way, work out which genes have been changed. I mean, a Mendelian way, work out which genes have been changed. And that's been an extraordinarily interesting area of science, understanding the domestication process that's underpinned this extraordinary increase in productivity in maize. Okay, so all of these extraordinary sources of genetic variation have allowed us as people the possibility of mixing and matching them by breeding to produce this kind of change in our crop plants. And then we run into a problem. Now we have this, which is a wonderful set of genes giving a wonderful set of properties that we like, but actually we'd like to add another property, a new one, like disease resistance, because there's a new disease. We find a lovely resistance gene in teosinte, but to get that teosinte gene into maize, we have to use this conventional breeding approach that's called introgression. So we would take our elite crop and we would take the wild relative, this is what the whole plant of teosinte looks like, and we would cross them together, and then we would have to keep back crossing them for generations, multiple generations and generations, because as soon as you cross them together, you've mixed up all of their genes, and there's only one gene that we really want to move, and so you have to take the children and cross them again to the elite parent, and take the children of that and cross them again to the elite parent, and every generation you can get rid of an increasing amount of the genes you don't want from the wild relative, and every generation you have to select to make sure that the plants you're keeping in the program include the one gene that you do want that encodes the disease resistant. And this is a very long and slow process. And so we've tried to figure out ways to speed this up over the years. And we've done deliberate random mutagenesis to try to increase the genetic diversity in the gene pool of the breeding populations we have for crop development. And then we have tried to invent ways to do deliberate horizontal gene transfer, which is how I would classify genetic modification. And so that way you can isolate the gene from the wild relative and just transfer it directly into the crop we're interested in, and that saves you all of those many, many back-crossing generations. So that's a tool that is now readily available to us and has actually been extraordinarily useful in the lab in understanding how genes function. It's absolutely crucial in figuring out which gene does what, the ability to move it around in a laboratory context. And as we know, it's been rather more controversial in applying it in an agricultural context, and I imagine we're going to have a good discussion about that later this afternoon. And that discussion has been kind of amplified by the fact that we now have a whole bunch of new techniques that allow us to make deliberate changes in genomes differently. I'm running out of time, so I'll fairly rapidly skip over the next two slides. But I mentioned earlier that there are a lot of repair mechanisms in genomes that deal with the fact that quite often DNA will break, and you will get these double-stranded breaks. And the double-stranded breaks are repaired by one or other of two mechanisms. Either the ends get stuck back together again, and this is called non-homologous end joining, or you can use the other copy of the DNA to repair it by a recombination process, and that's called homologous recombination. And both of those processes happen. And in fact, the ability to stick new bits of DNA into the genome essentially takes advantage of the fact that there are these breaks, because if you add a bit of DNA into a genome, it will have some probability of being incorporated at a point of a non-homologous end joining process. So here's a bit of DNA and it's going to get stuck in here. I'm going to ignore this. These exciting genome editing techniques that have come up recently that you may well have heard of provide a way to dictate where the double-strand break is. That's the core of them. There's all kinds of fancy stuff associated with it, but the key idea is you can choose where that break will be. And that gives you the extraordinary power both to choose where you might be able to put a new bit of DNA, but also to create mutations in specific genes in your organism of interest, because if you just have a break that gets repaired, there can be an error in that repair, and that will result in that gene not working anymore. So these tools are new, very powerful ways to change genomes, so we can have now deliberate targeted mutagenesis as well as the deliberate random mutagenesis we had before, and the precision of this kind of horizontal gene transfer can happen. So now, where are we in life? We have all these tools. We have a way to understand which genes do what by the genetics that Mendel discovered in the new molecular era. We have a whole range of ways to make on purpose variation in those genes that we can incorporate in plant breeding. And as I've described, evolution, natural selection, has given us quite a good chassis. It's given us a washing machine. It's a big white machine. It squirts water. It's very impressive, and it washes clothes. But actually what we want from a crop point of view is not a washing machine. We want a dishwasher, because natural selection has given us all kinds of annoying traits which are not very useful. If you want to wash dishes in the washing machine, you put them into the drum and bad stuff happens. So we need to understand which part of the washing machine is the round drum that goes round and round. We need to take that out and replace it with some slidey drawers, and then we'll be in much better shape. And we need this in the context of the real urgency of the kind of problems we're facing on the planet. So there's this kind of perfect storm triangle of energy, water, food in the context of climate change. All of these factors are very closely intertwined. So whilst plant breeding over the last 10,000 years has given us a pretty amazing dishwasher with relatively limited understanding of how the drawers work and how the drum works and the relationship between them, we now need a super efficient dishwasher that uses less water and less detergent and heats the kitchen. So we need to use plants much more effectively, and to do that we really do need to understand all those moving parts and think much harder about the way that we target them. And there are many things we might want to do. And there's a huge amount of basic plant science research driven at understanding all of these different processes, whatever they might be. And from a point of view of the policy and the public debate, I think a much more interesting question, which techniques should we be able to use to do this stuff, are these bigger questions. And these are the ones that people really care about. They care about what we should grow, how and where we should grow it, how it should be distributed, who should benefit and who should decide. People worldwide are much more interested in the social justice of the system than they are in the technologies that underpin it. And most people's concerns about the technologies are their ability to violate their sense of social justice. And so I think it's very important to have these debates in the context of understanding the science, of course, but in the place that people care about, which is much more the socioeconomic context in which they're being used than the techniques themselves. Thank you.